So here we're going to understand ion exchange chromatography, and chromatography is just simply a word that describes molecules being purified or isolated from a mixture using various techniques. And specifically, we're going to understand ion exchange chromatography, which is based off of the theory of charged molecules interacting with functional groups within the matrix of a column. So if we take a look at a column, something like this, which we will see in our labs, this column is made up of an inert matrix, which may be agarose, it may be polyacrylamide, whatever it may be, but we have these beads also which are inert. And we have functional groups that we bind onto these beads. Now, these functional groups may be negative or positive, it just depends on what molecules we are trying to isolate. For example, if I take negatively charged molecules. So remember, let's talk about proteins. So proteins are made up of amino acids, and amino acids can have various pKa values based off of which they can be protonated or deprotonated, so they may have a negative, neutral, or positive charge. So if I have these protein molecules with a negative charge and my functional group that is bound to this bead also has a negative charge, there will be no interaction between negative and negative because they will repel each other, and these negatively charged molecules will simply elute from the column. But if my functional group had a positive charge, it would interact with that negatively charged molecule and it would not elute out of the column unless we changed the pH of the column, because that would affect the protonation state of our molecule. So going back up here, we have an inert matrix beads, and we bind specific functional groups to the inert matrix based off of what we are trying to isolate, and those functional groups may be negative or they may be positive. And once we reach the pi, remember the pi is the point at which the net charge of our molecule of interest is going to have a charge of 0. Remember, we can have various functional groups and they can be negatively or positively charged, but when the overall charge is 0, that's when we say we have reached the pi. And now let's take a look at an example. So let's say that I have a mixture of molecules, and they are all positively charged because they exist in a buffer with a very low pH, and at a low pH, all the functional groups are protonated. So all the functional groups are protonated, we have an overall positive charge, we add it to the column, and the functional groups must have a negative charge so that negative and positive can form those ionic interactions. And we can see that the positives have bound to those functional groups. Now, the question is, once they are bound, how do we get them out of the column? And that's where we have to incorporate the concept of pi and increasing the pH. So if all of these original molecules exist at a low pH, they're all protonated. As we increase the pH, these molecules will become more and more deprotonated. And as they become more and more deprotonated, once we reach the pi, where they have an overall charge of 0, at that point they will begin to elute from the column. So let's say that these green molecules over here, they have a pi of 5. And now let's say I'm adding buffers of various pH values, and then I add in a buffer which has a pH of 5.5. So since the buffer that I'm adding has a pH of 5.5 and the pi, the isoelectric point of this molecule is 5, these green molecules will begin to elute from the column because we have passed the pi point. So now they not only exist as a net charge of 0, but they're moving towards having a negative charge. So we'll see these elute from the column. And let's say these pink molecules have a pi or isoelectric point of 7. Once I add in a buffer that has a pH anything greater than 7, let's say 7.3, I add it to the column. Now these molecules, they not only now have a charge of 0, but now they're moving towards a negative charge. And now they're no longer gonna have that ionic interaction because negative and negative repel each other, and they're gonna elute from the column, as we can see over here. And that is essentially ion exchange chromatography. It's based off of the charge of these molecules, and as we increase or decrease the pH of the column, they are going to elute from the column. And now it's really important to understand that in this example, we went over positively charged molecules and increasing the pH. Now this would be, and the functional groups were negatively charged, but this would be the same thing, but in reverse if we were using negatively charged molecules and a positively charged functional group. It would be the same theory, just in reverse. Once you pass the pi point, the isoelectric point of the molecule, it's gonna be eluted from the column. And in this way, we can isolate molecules based off of their charge. And that's exactly what's explained over here, where I said buffers with different pH values are added to the column, and the column buffers that we are adding will affect the charges on the molecule based on the pKa of the molecule itself, resulting in it being eluted.